Cellular respiration is a four-step process where glucose is broken down to create 38 ATP. Cellular respiration occurs in the mitochondria. The first step, glycolysis, occurs in the cytoplasm. The second and third steps, pyruvate oxidation and the Kreb cycle, both occur in the matrix. The fourth and final step, which is the electron transport chain, occurs in the inner membrane of the mitochondria. For glycolysis to begin, it requires energy. This energy is produced from the hydrolyzation of two ATP molecules. A phosphate group from the first ATP is then attached to the first end of the glucose molecule, creating glucose 6-phosphate. Glucose 6-phosphate then rearranges itself into its isomer fructose 6-phosphate. This is where the second ATP decides to join the party. This ATP also donates a phosphate group to the opposite end of the glucose molecule that the first ATP attached to, thus creating fructose 1,6-biphosphate. As you can see here, the original 6-carbon backbone splits into one D-hat molecule and one G3P molecule. D-hap is then converted into another G3P molecule by transferring the phosphate group from the first carbon to the third carbon. Now that there are two G3P molecules, we can now start generating energy. One hydrogen and two electrons are taken from G3P and transferred to NADH, allowing it to form NADH+. As a side note, NAD+ is a reusable coenzyme that functions at the active site of an enzyme. Its ability to accept and transfer both protons and electrons is what allows it to switch between NAD+ and NADH. While this is happening, G3P is also combining with the phosphate, which results in the creation of 1,3-biphosphoglycerate. A phosphate group from 1,3-biphosphoglycerate is given up to ADP, converting it to ATP. When the phosphate is removed, the molecule becomes 3-phosphoglycerate. By this time, 2 ATP have been formed, one from each G3P. Thus, the original energy investment of 2 ATP has been paid off. The 3-phosphoglycerate undergoes isomerization and becomes 2-phosphoglycerate. A proton and hydroxide ion are removed from 2-phosphoglycerate and combined to form water. After the removal of water, phosphophenylpyruvate, otherwise known as PEP, is produced. Since PEP is very unstable, it releases energy, enough energy to transfer a phosphate to ADP, resulting in ATP. Once the phosphate was removed from PEP, it becomes a pyruvate molecule. But if you remember back when fructose 1,6-biphosphate was split, it creates 2 G3P molecules, therefore resulting in 2 pyruvate molecules. Now this concludes the process of glycolysis. The pyruvate molecules can now enter the Kreb cycle. The second step of cellular respiration is pyruvate oxidation. With the help of a membrane carrier, the pyruvate molecules enter the matrix of the mitochondrion and pass through the outer and inner mitochondrial membranes. Once the coenzyme A attaches to the acetal group of the pyruvate molecule, it enables NAD+ to be reduced to NADH and lose its electron. CO2 is then removed as waste. The final product of pyruvate oxidation is acetyl-CoA. Acetyl-CoA is what initiates the next step in cellular respiration, the Kreb cycle.
Next up is the Kreb cycle, but before we go through the steps of the Kreb cycle, we need to know a bit about the background. So here are a few quick facts. The Kreb cycle occurs in the matrix of the mitochondria. This is different from glycolysis, which occurs in the cytoplasm. There are 8 reactions that occur in the cycle, and each is catalyzed by a different enzyme. For every 1 glucose that enters glycolysis, it must also undergo 2 cycles of Krebs. Up next, every step of the Krebs cycle will be outlined. Alright, so sit back, relax, and listen carefully. The Krebs cycle begins with acetyl-CoA, which is a product of pyruvate oxidation. The acetyl-CoA reacts with oxaloacetate to form citrate. The CoA is then removed. Isomerization happens to the citrate, and it becomes isocitrate. And here's a quick study tip to remember this step. Think isomerization creates isocitrate. Isocitrate is then oxidized to become alpha-ketoglutarate. Notice the NAD plus ion gains an electron to produce the coenzyme NADH. This happens through reduction. Step 4 involves alpha-ketoglutarate binding with CoA to form succedyl-CoA. Next, alpha-ketoglutarate is oxidized by NAD plus to create NADH. Also notice, carbon dioxide is removed as waste. During the 5th step of the Krebs cycle, CoA is lost, and this causes a large release of energy. Succinyl-CoA then becomes succinate. Phosphorylation occurs, and GDP is phosphorylated to become GTP. Next, a phosphate group comes in and converts ADP to make ATP. The next step involves an FAD ion coming in from the matrix to oxidize succinate and form FADH2. This results in succinate becoming fumarate. Water is added to the mix to transform fumarate into malate. Finally, there is one more redox reaction, and NAD plus becomes NADH and forms oxaloacetate. That has been a description of the Krebs cycle, but keep in mind that for every 1 glucose molecule that enters the cycle, it needs to go through 2 cycles of Krebs. And now for a quick overview. The Krebs cycle produces 6 NADH ions, 2 ATP, and 2 FADH2. And now we are done.
The electron transport chain is found in the inner mitochondrial membrane and is the final step of cellular respiration. It has 5 major components: complex 1, complex 2, complex 3, complex 4, and oxygen. The ETC's sole purpose is to create ATP through oxidative phosphorylation. NADH binds to complex 1, called NADH dehydrogenase. The mobile electron carrier UQ carries the electrons from complex 1 to complex 3, complex 3 also known as cytochrome BC. From there, the electrons are moved by mobile electron carrier cytochrome C from cytochrome BC, which is complex 3, to the complex 4, which is cytochrome oxidase. As you can see, FADH2 binds to succinate dehydrogenase, complex 2. Unlike the NADH molecule, FADH2 begins at succinate dehydrogenase, complex 2. FADH2 will make less ATP due to the lower amount of electron carriers it has to pass through. The hydrogen ions that have been pumped into the intermembrane space, as shown, combine with oxygen to create H2O. Oxygen is the final electron acceptor due to its strong electronegativity. It is the only molecule that is able to strip the last component of the ETC of its electrons. Once this process has finished, the ETC has created an electrochemical gradient to power ATP synthesis. Chemiosmosis is where ATP is made via oxidative phosphorylation. This process can only occur after the electrochemical gradient has been created by the ETC. Hydrogen ions begin in the intermembrane space and are pushed through the ATP synthase into the mitochondrial matrix. The force of the hydrogen protons creates a bond between ADP and phosphate, therefore creating ATP. The process of the hydrogen protons being forced through the ATP synthase to create a bond between ADP and phosphate to create ATP is known as oxidative phosphorylation. The net amount of ATP created throughout cellular respiration is shown. Altogether, 6 ATP are created through glycolysis, 4 of which are created by 2-FADH. Pyruvate oxidation creates 6 ATP through 2 NADH molecules. The Kreb cycle creates 18 ATP by the 6 NADH, and the 2-FADH2 create 4 ATP, resulting in a theoretical total of 36 ATP.